Okay, hi everyone. This is the lecture on chapter 10, which starts the new unit, right? Remember this new unit, unit 4. We're going to talk about cell injury, we're going to talk about inflammation, and then we're going to talk about altered cell growth and regulation. So chapters 10, 11, and 12. And so we're going to talk today about the first chapter, chapter 10. Okay, so these are the mechanisms of cell injury and aging. All right, so the first learning outcome is to talk about the difference between reversible and non-reversible or irreversible cell injury, talk about cellular adaptation, and just some concepts that are related to cell injury and aging. So if just a little bit of overview, okay, just to sort of a reminder of if we're going to learn about pathophysiology, right? We have to always think about what's happening on the cellular level, right? And so this unit really is specifically focusing on the cellular level, but really everything that we talk about in pathophysiology in terms of different systems that are affected, whether it be respiratory or cardiovascular, right? We're always gonna be coming down to that cellular level. So remember that cells are the smallest structural and functional unit of an organism. When we talk about the concept of cellular adaptation, basically that just refers to the changes that the cell will make in response to some sort of stressor, right? Now the stressor can be, a change in the ion concentration inside the cell, right? It could be a change in the ion concentration outside the cell, right? It could be a change in a certain protein level or something like that, right? There's various types of stressors that can occur, okay? And so these changes can be induced by an injury, right? Like a traumatic brain injury or by a heart attack or something like that, okay? And so we're going to talk about some of these different things as we move through this chapter. The overall goal for that cell is to maintain homeostasis, right? We've heard that term before. And again, homeostasis just in general means, steady state. So it is super important. And again, hopefully we got this from the chapter on, regulation of ion concentrations, right? And osmolarity of the blood and things like that, right? That cell needs to maintain homeostasis. And it's not only about what's happening inside the cell, but also in that extracellular environment as well, okay? So if the cell does encounter some sort of stressor, okay, it will try to adapt to that stressor, right? And basically, fight for its own survival by conserving resources, right? So it sort of decreases the amount of metabolic activity that's going on. It may decrease or even stop some of the functions that it might normally perform because, again, it's trying to survive, okay? So it's not going to be able to expend that extra energy performing a given function. And so that's what this last part says, focusing on their own survival, right? So the cells will adapt to some sort of stressor in the environment to basically be able to survive. And then hopefully once that stressor goes away, they'll be able to regain normal function, okay? And so if this cell is able to regain normal function, right, it would be reversible. If we talk about irreversible or non-reversible, that injury or that stressor is going to cause changes in the cell that are severe enough that the cell is not able to regain function, okay? So if we look at the figure on the next slide, this is basically showing you the difference between reversible and non-reversible or irreversible injury, okay? So here's your normal cell. Again, remember it needs to maintain homeostasis. That means it needs to keep levels of proteins and enzymes at that appropriate level so that, metabolic reactions can occur. It needs to maintain appropriate ion concentrations, both inside and outside the cell. It needs to maintain the appropriate amount of water, right? These are all things that are part of homeostasis. Okay, so some sort of injury can occur, right? And it could be like a transient sort of mild type of injury that cell encounters, right? And so that induces some sort of stress on that cell. The cell will try to adapt, okay? However, if that cell is not able to adapt, so if it's able to adapt, right, it'll just go back to its normal function, okay? Again, if you look here at this injury here and the cell is not able to adapt, right, and you have sort of this progressive, more severe type of injury. Again, cell tries to adapt, not able to adapt. It then is not able to regain normal function. We call that an irreversible injury. Ultimately, that cell will end up dying. And there's two sort of different types of cell death that we'll talk about at the end of the chapter: necrosis and apoptosis. Okay. So again, think about, if you think about organ function, if enough cells within that organ die, the organ will ultimately fail. Okay. Or if you just want to think about tissue, right? If enough cells of a certain tissue die, right? Again, that tissue is not going to function properly. Okay. And so, and different organs, different tissues can tolerate different levels of cell death, right? But bottom line is, if too many cells are dying, obviously it will lead to organ failure and then potentially death. Okay. So anyway, cells can be injured. Cells have processes to try to adapt to that injury, and those can be reversible. But sometimes the injury, the stress is severe enough that it is non-reversible. Okay, so what are some concepts that are related to cell injury and aging? Again, just like I mentioned, right? Injury to cells or aging cells that end up dying, or if enough cells end up dying, it affects the function of the entire body. Okay. So again, this is why it's so important to understand these mechanisms and these processes in a pathophysiology course. So if you want to kind of just talk about some concepts that are related, and we'll look at the figure in a second, cellular regulation, okay, inflammation, oxidative stress. These are all concepts that are related to cell injury and to sort of normal aging of the cell. Okay. Example here, right? If we think about heart cells, myocardial cells, they will sustain an injury, right? When they're deprived of oxygen, obviously. Okay. That injury to the cells is reversible if that blood supply is only compromised for about 10 to 15 minutes. However, if the blood supply is compromised for a longer period of time, it will be irreversible. Those cells will die. If enough myocardial cells die, that will cause the heart to stop working, and you can have a massive heart attack, and you could die right on the spot, right? If only a few of those cells die, maybe not such a big deal. You could also kind of, have an ischemic event or have a minor heart attack where you have a deprivation of oxygen, but those cells end up regaining function, right? So important that if there is a blockage, right, you get to the hospital right away, they put a stent in or whatever and open up that blood vessel to regain that oxygen supply. All right, so if you look at figure 10.2, again, this is a figure that we see in every chapter, just showing you concepts that are particularly related to here, cellular regulation, right? And then we talked about inflammation and oxidative stress. And then again, if we talk about cellular regulation in general, I mean, that's really a general term, right? I mean, it's kind of any particular, function of the cell will be altered if the cell isn't regulated properly. Okay. If you then want to look at some concepts that are related to it, oxygenation, I just gave you an example really, of something that involves both perfusion and oxygenation, right? In terms of the heart attack. Also, you can think about stroke, right? This is going to affect brain tissue rather than heart tissue, okay? We'll talk about these, hypoxia, hypoxemia, and dysoxia. We'll talk about that. But again, and we'll talk about carbon monoxide poisoning specifically. But again, all of these things are pertaining to oxygenation, okay? If you talk more specifically about metabolism or immunity. So immunity here, this is referring to like autoimmune diseases, like an example, myasthenia gravis, which I also will mention in the lecture as well. If we talk about problems with metabolism in general, particularly here, for fatty liver, it's problems with metabolizing lipids, particularly a lot of that happens in the liver, right? That happens in the cells in the liver. Remember we talked about this when we talked about alcoholism, right? So chronic alcohol intake leads to something called fatty liver because the liver is metabolizing the alcohol and therefore isn't able to metabolize those fatty acids, or those lipids. And so it leads to this fatty liver. So again, but you can have other problems with metabolism that might result in things like that, okay? If you talk about inflammation, oxidative stress, these are some other sort of secondary concepts or diseases that are related to that. And we'll talk about some of these in later chapters, okay? And so that's pretty much the gist of it there. Okay, so this next section, we're just summarizing cell structure and function, but then also talking about consequences of injury to certain organelles, okay? The first part should be review. So if we're talking about pathophysiology, we're talking about the human body, right? We're talking about eukaryotic cells, okay? So eukaryotic cells have a true nucleus. They have other membrane-bound organelles. We're not even going to talk about prokaryotic cells, but you are talking about prokaryotic cells a lot in microbiology, right? So in microbiology, you focus on, bacteria, which are prokaryotic cells. So obviously those prokaryotes can greatly impact the human body and impact eukaryotic cells, but we're going to focus on function of eukaryotic cells here. So primary components, again, this should be review, okay? Nucleus obviously houses the genome, right? DNA, RNA, DNA replication and transcription happen inside the nucleus, okay? So these are particular things that we're going to particularly talk about, I guess, right? So cell membrane, we talked about the cell membrane, again, a lot in the chapter where we're talking about ion imbalances, right? And we talked about, hypertonic and hypotonic and isotonic sort of external environment, right? And what that means in terms of water getting through the plasma membrane, okay? So all of these things are important, right? So the cell membrane is super important because it's going to allow certain things to get in and certain things to not get in. And so that really regulates the internal environment of the cell, but also the external environment as well. Cytoplasm, that's basically where all of these organelles are housed. And it is sort of gel-like, but it is, things can move around in that cytoplasm. Okay, so again, I'm not going to put a cell diagram on your exam and say, can you identify the ribosomes? We should know this. If we don't, that's a problem. You should review that on your own, but you should be able to you should already be familiar with these different organelles that are labeled here, okay? One thing I think that's now sort of a little more appropriate for this course and sort of an extension of just understanding basic function of some of these main organelles is now what happens when these organelles do not function properly, okay? So in this column here, you have just basic functions of each one of these organelles, right? And what happens if there is a dysfunction, okay? So we're talking about, sort of cell injury, right? Or cellular stress and the idea of that, what happens as a result of something going wrong inside the cell, okay? So here we're particularly focusing on, well, what happens if the ribosomes are not functioning properly, okay? These are generally, so ribosomopathies, right? Generally caused by genetic abnormalities. Ribosomes synthesize proteins, right? So if there is a dysfunction of the ribosome, proteins will not be synthesized properly, okay? And can lead to, obviously, disease. And it'll be specific in terms of which proteins are particularly affected, okay? If you talk about the endoplasmic reticulum, okay, it says here, interferes with pancreatic digestive enzymes, liver proteins, glycogen storage, metabolism of lipid-soluble drugs. Okay, the ER is involved. Now, remember, there's a smooth ER and there's a rough ER. If you talk about smooth ER is involved in lipid synthesis, but also lipid metabolism. And so that's why you'll see if there is a dysfunction of the ER in general, or particularly the smooth ER, right, it's going to affect metabolism of lipid-soluble drugs, okay? If you talk about the Golgi, okay, one thing that will be affected if you have dysfunction of the Golgi is secretion of hormones. Remember, especially in particularly, I guess, think of like peptide hormones. When proteins or peptides are produced in the ribosome that sit on the ER and the rough ER, they ultimately end up getting packaged and sort of identified in the Golgi complex. And so they go to the Golgi complex, and then they would be secreted out of the cells there. And so if there's something wrong at that level, the secretion of hormones, of these particular types of hormones, could be affected. Talk about mitochondria, okay? If there's an issue with the mitochondria, you're going to have a decrease in cell energy. Why? That's where ATP is produced, okay? Says here, leads to cell injury or death. Yeah, that's a major issue, right? And it says it can cause whole systems to fail. If cells do not have energy to perform the function that they need to perform, yes, you're going to have really serious consequences. Lysosomes, these are kind of like the digestive system of the cell. And so there will, and peroxisomes too. So lysosomes and peroxisomes are sort of similar, but they kind of just break down different things. And so, things will get broken down, old organelles, maybe a bacteria might come into the cell, right? The cell will take it in like a white blood cell and break it down. And so that's the job of lysosomes and peroxisomes. And so if they're not functioning properly, either an enzyme inside the lysosomes or the peroxisomes is not functioning properly, it leads to buildup of things, okay? So a lysosomal sort of storage disease, one example is Tay-Sachs. For peroxisomes, again, you'll end up having an accumulation of fatty acids, and again, can lead to cell death. And so a lot of times, with peroxisomes, this is kind of a little bit related to kind of the reactive oxygen species concept, or the oxidative stress. So there are enzymes, or if you think about fatty acids being broken down in the peroxisomes, they then produce hydrogen peroxide. And then there's another enzyme that will break down the hydrogen peroxide. But you'll see in one of a couple slides when we talk about reactive oxygen species, that hydrogen peroxide is one of those reactive oxygen species. So if the cell is not able to get rid of the hydrogen peroxide, that leads to major problems. It damages a lot of the molecules in the cell, okay? So anyway, there are a lot of diseases associated with lysosomal dysfunction, and we call them, again, usually lysosomal storage diseases, because you get accumulation of material in the cell. Once enough accumulates, it leads to dysfunction and can ultimately lead to cell death. Okay. All right. So let's specifically talk about cell membranes, right? And so issues that are that arise when there is some sort of dysfunction of the cell membrane. Okay. So again, primary function of the cell membrane is that it's semi-permeable. Okay. It allows some things to get through, other things cannot pass through. It's there to separate that intracellular and the extracellular environment, okay? If you think about proteins that sit within that cell membrane, okay, there are channel proteins. So again, we spent a whole chapter talking about ion imbalances. What happens if you have a sodium imbalance? What happens if you have a potassium imbalance? The way all of those ions that we talked about enter the cell or exit the cell are through channels, okay? So if there is a dysfunction of a channel protein, it likely affects a certain ion, okay? So there are specific channels for chloride ions. There are specific channel proteins for sodium ions, okay? And as you guys know, if ions or the concentration of ions is not maintained properly, that also interferes with the concentration of water, okay? Water itself also has its own specific channel. And again, I think I mentioned this in chapter 9. Not chapter 9, I guess that was chapter 8, right? And that channel is called an aquaporin, okay? So it says here, so there are channels, ion channels, that just normally are there to maintain proper levels of ions, right? Whether it be sodium or potassium. There are also specific ion channels that are involved in generating action potentials in nerve cells and muscle cells, okay? So again, yes, they'll move ions across the membrane, and that's what allows an action potential to be generated, right? That's what allows muscle contraction. That's what allows neurons to communicate with one another, to basically, participate in any function you can think of, any brain function, okay? There are also receptors that are linked to channels. So when something outside the cell binds to that receptor, it opens a channel. That then may cause an action potential and activate a neuron or a muscle cell, okay? So again, we should be aware of this basic information. And if we're not, then that means you need to go and read about it a little bit more, okay? We should know about ion channels. We should have heard about ion channels before. Ligand-gated ion channels and voltage-gated ion channels. That's sort of what I was referring to here. So if you need to, it may be helpful to go back and review that. I know there's information about that in a basic anatomy and physiology textbook. So anyway, I expect that you know that you sort of have that basic knowledge. Okay. So anyway, there are many different types of channelopathies. Okay. So meaning what happens if these channels, some of these channels that I mentioned, right, these ion channels are dysfunctional. Okay. So a channelopathy is basically any disorder that's caused by dysfunction of a channel protein. Okay. They can be caused by genetic mutations, but they also can be autoimmune related, right? So the body is then going to attack that channel. Okay. So lots of examples here. I actually added in some more examples. So sorry, it's kind of so squished together. I just didn't want to end up moving on to another slide. So here are some examples, right? A pain channelopathy, meaning if we talk about receptors, pain receptors, okay? If there is a dysfunction there, it can lead to hypersensitivity of pain, but it also can lead to disorders where you actually can't feel pain at all. Okay. Cardiac channelopathy. Actually, some of these cardiac channelopathies have been linked to sudden infant death syndrome. Okay. Myasthenia gravis is an example of an autoimmune, right? So I have written here acquired. So in myasthenia gravis, ion channels, okay, that respond to acetylcholine are destroyed. So again, it says ion channels / receptors. So the acetylcholine receptor itself is an ion channel, okay? So when acetylcholine binds to the receptor, it causes opening of that channel, okay? If that channel can't open, right, it leads to a dysfunction of those muscle cells. So in myasthenia gravis, you see progressive muscle weakness, ultimately leads to respiratory failure because the diaphragm will be affected, okay? More examples. If you talk about a renal channelopathy, diabetes insipidus, we mentioned this again in a previous chapter, where you have decreased responsiveness to ADH, okay? That results in a loss of large amounts of water in the urine, okay? A respiratory channelopathy, or there are respiratory channelopathies associated with asthma and cystic fibrosis. So in asthma, calcium channel affected. And so if the calcium channel is affected in smooth muscle cells, right? If we talk about airways being constricted, right? If you want those smooth muscle cells that line those airways to be relaxed, but if they're contracted, they cause constriction of that airway. In cystic fibrosis, it's a chloride channel, okay? So lots of examples that I gave you, right? I expect that you should be able to give me an example or two if I ask you that on the exam. Okay, so this is just showing you a picture of the cell membrane, right? So remember, the plasma membrane is made up, primarily of a phospholipid bilayer, right? But there are lots of proteins that sit within that membrane. We were particularly focusing on channels, and here's an example of a channel protein. Okay, also cholesterol is within that membrane. Cholesterol is involved in maintaining the fluidity of the membrane. This is just pointing out a transmembrane protein. So a transmembrane protein will span both bilayers. A peripheral protein sits on the outside. Well, so I say on the outside of the bilayer, but towards the inside of the cell. Okay, so again, we should already know this. We should be familiar with this. Okay, glycoproteins, right? There are also glycolipids, and all that means this is a protein that has a carbohydrate chain attached, and these have specific functions as well. Okay. Again, I can't go through all of this now. This is, again, basic knowledge that we should already have. Okay, Now, if we talk about things that are particularly related to cell metabolism, right, or energy metabolism, okay? If we just say metabolism in general or energy metabolism, we're just talking about the production and the utilization of energy, right, or ATP within the cell. The cell uses energy to make carbohydrates, right, make amino acid, or to make proteins, right, lipids. Also, the cell can break down these molecules as well, okay? So you kind of have two main types of metabolism. You have catabolism, which is breaking down, which I just mentioned, right? So breaking down stored nutrients and I don't know why it really says body. I don't know if I would say body tissues, but I guess that's true. But think about molecules, right? So the cell has the ability to break down proteins. The cell has the ability to break down larger carbohydrates, right? And then can get energy from the breakdown of those molecules. So let me just take that. I don't really like what that says there. I'm just going to leave it like this. Breakdown of stored molecules. Okay? So again, it's in general, you're thinking about breakdown of stored molecules, larger molecules. In anabolism, this is when the cell is making something, right? So you're making proteins from amino acids. You're making complex carbohydrates from more simpler carbohydrates, okay? These types of reactions, anabolism or anabolic reactions, will require energy, okay? Energy has to be put in, you're making something. If you have any abnormalities in cell metabolism or energy metabolism, in the ability of the cell to produce ATP, like I mentioned in that table, it will have catastrophic effects on the cell, okay? Usually leads to cell death, and again, ultimately can lead to organ failure if enough cells die, okay? The next thing that I want to talk about is cell-to-cell interactions and connections, okay? And sort of what this is involved in. So cells need to be able to interact with one another, right? If you think about, so it says here that it's critical to the development and the function of multicellular organisms. So if you even just think about, during development in general, right? If we think about, the embryo becoming a fetus, right? There is so much communication between cells that's necessary for that organism to develop properly, for organs to develop properly, okay? If these cells aren't able to interact with one another and essentially talk to one another, development will not occur, okay? Now, that communication and that interaction between cells still happens in the adult as well, right? So if we're talking about just the function of the human body, super important as well. So on the next slide, there is I'll talk a little bit about cell junctions. I'll show you what I mean by that. But basically, a cell junction is basically a connection between two cells, right? So a stable interaction or a stable connection between cells. It says here, communication and organization of cells within a particular tissue. So if you think about endothelial tissue or epithelial tissue, those epithelial cells are joined together by cell junctions, right, to create that tissue, okay? Remember, when you think about epithelial tissue, right, it'll line organs, okay? And it sort of creates a barrier between sort of inside of that organ or let's think about, I guess, blood vessels or GI tract, right? You have epithelial cells that will line the GI tract, right? And anything that needs to get into the bloodstream has to go through those cells, okay? They have tight contacts between one another. And again, that forms the tissue. It says chemical and electrical signals. Also, the junctions between these cells can allow for chemical and electrical signals to pass from one cell to another, okay? There are also more transient types of cell-to-cell interactions, and this usually involves immune system cells, and we'll talk about that a little bit more in chapter 11. If you lose communication between cells, it can lead to uncontrolled cell growth, and we'll talk about that more in chapter 12, okay? Let's look at 10.5, and then I'll come back and talk about extracellular matrix. So if we look at figure 10.5, this is what I was talking about, these junctions between cells, okay? And so it's kind of showing you different types of junctions between cells. We don't have to worry about different types of junctions, I guess, for right now. but know that there are different types of sort of connections between cells. Okay, if you talk about a tight junction, think about the example I gave before about, in the GI tract. Okay, you need to make sure that things can't just squeak through between cells, they have to go through the cell, right? So anything that's going to end up getting to, say, the blood has to go through this cell, and then it's a regulated process. If things can leak through, then there's no regulation at all. Okay, these gap junctions that it's showing you here particularly will allow ions and molecules to flow between cells. Okay, you don't always see these gap junctions. It's in certain cells, certain cell types, and certain tissues. Okay. All right, so let's go back and talk about the extracellular matrix. So just as it says, it's on the outside of the cell. So it says non-living material that's secreted by cells. So cells themselves, which are living, will secrete proteins that are part of the extracellular matrix. Okay, so it provides structural support to cells and tissues. If you think about, connective tissues, okay, then you probably, it's kind of easiest, I guess, to think about that when you think about extracellular matrix, right? So if you think about bone as being a really hard and structured type of tissue, it has a really strong extracellular matrix, okay? Blood, on the other hand, is a more fluid extracellular matrix, okay? Even though the extracellular matrix is outside of the cell, and it itself is non-living, it still plays a role in regulating cell behavior, okay? So what happens if there is some sort of protein that's altered, that's part of the extracellular matrix, right? Or some other dysfunction, it can lead to dysregulated or deregulated cell proliferation, right? And invasion, right? So basically, if you think about metastasis, right? Cancer cells or that can detach from one area and then move somewhere else, right? Because there's a dysfunction of that extracellular matrix. Those cells are not staying anchored in the tissue that they belong in, okay? And so this, a lot of dysfunction of the extracellular matrix is related to cancer. Also congenital defects as well, because, if you think about the formation of tissues and organs during development, there is a lot of cell migration that has to occur. Cells need to go to the right place, right? And that is dependent on the extracellular matrix as well. And so if that doesn't happen, it obviously can lead to congenital defects. Okay. So for extracellular matrix, you should understand what dysfunction of the extracellular matrix leads to, right? It's going to particularly affect these processes that are listed here. Okay. So again, it says failure of cell death. So again, when you think about cancer cells, these are dysfunctional cells that should go on, to die, but they don't, right? And they then create more and more problems. Also, cancer cells are in a non-differentiated state, meaning they have this sort of stem cell-like characteristics where they keep dividing, okay? And they actually don't provide the function that they need to for that specific tissue, okay? The second half of the lecture, we talk a little bit more about some of these things, okay? All right, so if you look at figure 10.6 here, this is just showing you this extracellular matrix, right? So I gave you the example kind of talking about epithelial tissue. What this is showing you here, so is that this is kind of this extracellular matrix part, right? And if you look, there's a bunch of different types of proteins that make up that extracellular matrix, right? So it's showing you the basement membrane here, basement membrane. So this is where the epithelial cells attach, kind of, right? And what anchors them into the tissues, right? We just kind of talked about it. If there's a dysfunction in that extracellular matrix, often cells will sort of break away from their tissue and move somewhere else where they don't belong, right? So that's what this is showing you here. These guys are anchored to what we call this basement membrane, and it's composed of all these different, proteins. So the extracellular matrix is a major component sort of in the formation of tissues, okay? And so again, this is now showing you some of the proteins, or right, showing you these integrins and these other proteins that are part of the interstitial matrix, right? So interstitial fluid, remember that matrix between cells here. This is particularly just showing you this basement membrane where we think about cells kind of being attached to, right? It's particularly if you think about epithelial cells, they're usually anchored to one part, right? And then the other side of the cell, say here, would be facing, say, towards the inside of the small intestine or something like that, right? Okay. Again, do you need to like memorize anything that's in this figure? No. Okay. This is really meant just to be an example here to show you what we mean by extracellular matrix. Okay. So it is composed of different types of structural proteins. Okay. These proteins, and they all interact with each other and sort of provide this framework and again, and this structure for cells. Okay. And they are in the extracellular matrix basically is necessary for tissues to form, right? Okay. All right. Another concept that we need to talk about is cell signaling. Okay. And obviously we're just going to scratch the surface on this. But it's the idea that, you know, a hormone or any drug, a pharmaceutical drug, a neurotransmitter, right? In order for it to have its action on the cell, it has to bind to its receptor, right? And so, you know, I probably should have put another picture here. One second, I'm going to pause it and I'm just going to add another additional picture here to help. All right. So just to, I just added in a figure here just again, this is something that we should be familiar with or should kind of remember from a basic science course. So in terms of, you know, cell signaling, what do we mean by that, right? So remember, there's different types of signaling. I just want to kind of give you guys an example and to kind of conceptualize what's going on here. So, you know, think about, again, a cell can release a signal, right? A protein, a hormone, a peptide hormone, another type of hormone, a steroid hormone. In order for this to initiate its effect, right, it has to find its target cell and then bind to its receptor on that target cell, okay? And so sometimes, you know, if a cell releases something, it could then just affect those neighboring cells, right? So that's kind of one type of signaling. If we talk about neurons, right, and neurotransmitters, they will be released at the axon terminal, right? And then they can bind to that postsynaptic cell, very localized, okay? Again, they're going to bind to a receptor here. This picture's not great, but I think it's good enough to show what we need to show. So, right, it will bind to its receptor here on that postsynaptic cell. If you're talking about endocrine signaling, right, hormonal signaling, the hormone, this here is probably more related to like growth factors and things like that. Okay, you can see here I listed this as one of the types of first messengers. So growth factors will be released and they usually will affect neighboring cells here. Okay, for hormones, those are released into the bloodstream, right? And then they will again ultimately reach their target cells. If it's a steroid hormone, the receptor actually might be inside the cell, right? Or if it's a peptide hormone, it would bind to a receptor sitting on the surface. Okay. So again, just to remind you about that. So this is giving you, we're talking about first messengers, meaning it's the hormone, it's the neurotransmitter or the growth factor that's going to bind to its receptor. Also, right, pharmaceutical drugs. You guys are all going to take a pharmacology course. Or if you think about pharmacology, in order for a drug to have any sort of action on the cell, it has to bind to its receptor. Okay. The other thing you want to think about when you think about signaling is something called a second messenger. So let's just think about those in general as molecules inside the cell that will be activated, right? When the ligand binds to its receptor. And I have a picture, or the next slide will show you a little bit more about that, okay? Now, there are abnormalities in cell signaling that can lead to disease, okay? For instance, right, cells can lose the ability to respond to these external signals, okay? Cells can lose the ability to communicate with one another, okay? An example, type 2 diabetes, right? Target cells are unable to respond to insulin and bring glucose into the cell. There's plenty of insulin being produced, but it's about the cell signaling that's defective, okay? Cancer, cells will grow and divide, again, because they are unable or lose the ability to respond to inhibitory signals that exist in that extracellular environment, okay? So let's look at that next slide, okay? So figure 10.7 here. So this is kind of showing you a couple of different things here. So again, we talked about extracellular matrix, right? If you look at these here, these integrins, these are integrin receptors. And so these receptors will actually interact with proteins that are in the extracellular matrix, okay? And so these are involved in setting up those cell-to-cell interactions that we talked about, okay? You can also see, though, that these integrins are then also sort of indirectly, or let's say they're also linked to the cytoskeleton, so inside the cell. Okay, so binding of something to this integrin receptor out here will affect processes inside the cell. Okay, so that's what this is saying, cytoskeleton mediated signal. So particularly, it will affect things that are related to the cytoskeleton. Okay. If you talk about, also cells, hormonal factors and things like that, hormones, neurotransmitters, right, binding to the receptors, it will initiate some sort of signal transduction pathway inside the cell, which ultimately can affect, transcription or things in the nucleus. That's why everything's kind of pointing to the nucleus here. Also, growth factors I mentioned, right? They can bind to the receptor, same thing. They'll activate a whole bunch of things inside the cell, which we're leaving out here because beyond the scope of this class. But ultimately, all of these things that happen here are the link between the binding of the growth factor or the hormone to its receptor and what effect that's going to have, right? So some of the effects that can happen are proliferation, differentiation, protein synthesis, right? Attachment, meaning attachment to the extracellular matrix, migration, right? Shape change. All of these are things that can happen as a result of this first messenger binding to its receptor. But a whole bunch of other things need to happen inside the cell before this effect can occur, right? So if you have a dysfunction in some molecule that's involved in the pathway here, that's going to be just as important as having a defect in the receptor, right? Because you need all of these different steps that we're sort of leaving out, or these internal pathways, in order to have these effects down here, okay? So super important. These cell signaling pathways are super important. And there are many different specific cell signaling pathways that exist, okay? And again, we're not even gonna talk about that. Okay. So let's talk a little bit about some common causes of cellular injury. Okay, so what are some common causes? So here's a table that lists a whole bunch of different types of hypoxia. Okay, and so what do we mean by hypoxia? Low oxygen in the tissues. What do we mean by hypoxemia? Low oxygen in the blood. And then ischemia is a lack of blood flow. Okay, so hypoxia, right? Basically that the oxygen level is below normal, so less than about 80 millimeters of mercury. And this can be due to environmental reasons or because of respiratory disease, okay? So this is, again, if the oxygen levels of your blood are too low, this then will lead to your cells not getting enough oxygen, right? And we talk about ischemic hypoxia. The reason why your cells or the tissues don't receive enough oxygen is because there is decreased perfusion to that tissue due to some cardiac or vascular disorder. Okay, so think about heart attack, right? Anemic hypoxia, okay? This is caused by the oxygen carrying capacity of the blood being reduced. So conditions like anemia, carbon monoxide poisoning, these decrease the oxygen carrying capacity of the blood. And again, if there's not enough oxygen being carried around in the blood, again, there will not be enough oxygen delivered to the tissues and the body's cells. If you talk about histotoxic hypoxia, right? In this case, the tissue or the cells within that tissue are unable to use oxygen, okay? Because it's been poisoned. So for instance, like cyanide will actually prevent mitochondrial enzymes from using oxygen. So the oxygen is there, the cell's not able to utilize it. If mitochondria is not able to utilize oxygen, there will be not enough ATP produced. Okay. Also, hydrogen sulfide will cause histotoxic anemia. Okay. So just a reminder, right? Again, do you need to memorize any of this stuff? No, but we should kind of be familiar with this. This should ring a bell. If we're talking about the generation of ATP in the mitochondria, right? This is showing the mitochondria, right? It requires oxygen. You will not generate 36 to 38 ATP without oxygen, okay? So that's why these any type of hypoxia is really, really dangerous for the function of the cell, okay? Or really dangerous to the cell. Okay, so again, just to continue on here, right? If the cell does not have enough oxygen, it will severely compromise the amount of ATP that's produced. I mean, if there's no oxygen, the cell's not going to last very long, okay? And it will have widespread effects, right? Because again, ATP is used in the cell for basically every process that goes on there, right? So one example, sodium potassium pump. Again, I think I mentioned sodium potassium pump when in our ion imbalance chapter, right? But the sodium potassium pump sits on every cell and is important in maintaining proper membrane potential, but also, the concentrations of sodium and potassium, okay? And so every cell is always running this sodium potassium pump. And so obviously that requires ATP. So if there's not enough ATP around, this will not be able to happen, okay? Now, a lack of oxygen is reversible, again, to a point. But if that oxygen is deprived for a long enough time, or, it's severe enough, it may be irreversible. Okay. So if you think about different types of tissues and their sensitivity to hypoxia, right? So here I've listed here as sort of survival times. So the brain can only go, it's less than 3 minutes without oxygen. Okay. So if the brain tissue is deprived of oxygen for 3 minutes, cells are going to die. Okay. Kidney and liver, though, can survive about 15 to 20 minutes without oxygen. Okay. Skeletal muscle, 60 to 90 minutes. Okay. It says here can increase glucose uptake by 600%. So to compensate for that lack of oxygen. Bladder actually can go several days, okay, because it can use anaerobic respiration to generate up to about 60% of its energy requirement. Okay. So, there are compensatory mechanisms that can occur within the cell to compensate for a lack of oxygen. But if there is no oxygen being supplied to the cell, right, for a certain amount of time, then the cell's going to end up dying. Another term here. So dysoxia. Okay. So dysoxia is an imbalance between oxygen supply and demand. Okay. So it says tissues cannot make full use of available oxygen. Or there's a decreased oxygen supply. So you'll see this dysoxia in, if you talk about respiratory disorder, right? Pulmonary edema, asthma, drowning, cardiac disorders, heart failure, atherosclerosis, anemia, okay? And again, it's the idea that there's an imbalance between oxygen supply and demand, okay? So, I guess you can kind of think of like maybe during normal states or kind of resting state, the cell might be okay. But if there's an increased demand for oxygen, with some of these types of disorders, the cells will not be able to meet that demand. Okay. So that's what this is the idea here. So if you think about even other types of, so that's why it says here, it's sort of specifying the pathological part, because again, there's always going to be, I guess there'll be times when the cell is challenged, right? To be able to, and have to increase its oxygen supply to meet that demand. Okay. But again, if there is some sort of pathological process going on and the cell cannot meet that demand, then, it's called dysoxia. And so even bacterial sepsis, right? Physical agitation, seizures can lead to actually increased oxygen demand. Okay. And so if the cell can't meet that oxygen demand, it will then, can then lead to cell death. Also, we got to talk about oxidative stress again. Now, we mentioned oxidative stress, I think, even back when we were talking about obesity, and I think maybe a couple other times as well. So we'll talk a little bit about it in a little more detail here, okay? So again, this is another cause of cell injury, okay? So remember that this is kind of where we are. Common causes of cell injury. We talked about hypoxia, right? Different types of hypoxia. Dysoxia is another cause of cell injury, okay? And dysoxia really has to do with the imbalance between oxygen supply and demand. So it's the idea that there can be some sort of states where they will increase demand so much that the cell can't meet that demand, okay? Before we were talking about with hypoxia, this sort of reduced availability of oxygen, okay? Here, it's the idea that it can either do with supply or demand, and it's kind of the balance between the two, okay? So that's sort of the difference between dysoxia and hypoxia. Again, oxidative stress, another thing we need to talk about, okay? So imbalance between the production of reactive oxygen species, right, free radicals, and the ability of the body to counteract or detoxify those free radicals, okay? So again, let's talk more about this. So reactive oxygen species are ROS, right? These are oxygen-containing molecules that, again, include free radicals. They have this unpaired electron in the outer shell, which makes them unstable, okay? And then those unstable molecules can go on and damage DNA. They can damage other proteins within the cell. They can then induce the cell to produce even more free radicals. So they have this sort of waterfall kind of effect, okay? It says maybe associated with cell and tissue damage. There's definitely, I think, a lot of evidence that suggests that. They are produced by normal metabolic activity in the cell, but they can also be produced by external factors like ionizing radiation, also giving supplemental oxygen, and then reperfusion, meaning there was some sort of hypoxic event or injury that blocked blood flow, and then when that reperfusion happens, there seems to be a higher production of these free radicals, okay, or these reactive oxygen species. Now, antioxidants, right, will inhibit the production of these reactive oxygen species, or they will help to neutralize those and get rid of those that are already there, okay? I'm sorry, I doubled up on that there. So there are enzymes, okay? And we'll mention some of these on the next slide here, right? So SOD, superoxide, let me tell you what that is. That would probably be helpful, huh? Dismutase, I believe. Let me just make sure I'm not saying the wrong enzyme there. SOD. Yeah, superoxide dismutase. Let me just see if I have it in my notes. Let me just add it in there. I guess I don't. What happened here? Where are my notes there? Hello? Uh-oh. Sorry, guys. 1 second. So let's just add that in there. I did add reperfusion in there. SOD is superoxide dismutase. I'll probably spell it wrong. Superoxide dismutase. Okay, so this is an enzyme that's involved in basically sort of neutralizing those reactive oxygen species. Okay, catalase is another one. Vitamins A, E, C, also micronutrients like zinc, things like flavonoids. I think I spelled flavonoids wrong. Oh my goodness. Let's double check that I didn't spell that wrong. Oh no, I spelled it right. Okay, I think. Okay, so all of these things that are mentioned here are going to act as antioxidants, and they are going to help neutralize these reactive oxygen species or just inhibit the production of them to begin with, okay? So if we look at figure 10.10 on the next, it's not the next slide, it's the slide after that. Let's first look at 10.9, okay? So this is showing you, a little bit more detail in terms of these reactive oxygen species. Now remember, the cell itself produces these sort of normally from some normal metabolic processes, but certain toxins, radiation, reperfusion, these are all going to induce the number of reactive oxygen species and sort of create free radicals that normally wouldn't be there, okay? And so like I said, there are enzymes that exist to get rid of these reactive oxygen species, okay? So these are all different examples of reactive oxygen species, okay? So if you're talking about superoxide here, right? This superoxide dismutase can basically get rid of that, okay? It'll convert that into hydrogen peroxide. There are specific enzymes like catalase that will basically remove hydrogen peroxide, okay? So both superoxide, hydrogen peroxide, they are toxic to the cell, okay? So if those molecules exist in the cell, they will damage other molecules, just like this hydroxyl radical here. So again, if these aren't gotten rid of by these processes that are showed here, they ultimately will lead to the production of hydroxyl radicals, which will lead to DNA damage, right? Can actually cause mutations in the DNA, can cause effects on lipids, right? So lipid peroxidation, particularly if you think about phospholipids that make up the plasma membrane, it can destroy those, okay? And also can lead to protein modifications that will lead to dysfunction of enzymes, right? So can actually cause misfolding of proteins, right? So you can see how these are pretty nasty in terms of what they can do to the cell if they're not taken care of, okay? So one particular type of vitamin, right, that is involved in getting rid of these free radicals is vitamin E. So vitamin E is really important. It's a lipid-soluble antioxidant, and it's particularly important in neutralizing those free radicals that would affect those phospholipids, okay? And so you can kind of see here, here's your vitamin E. It will basically grab this free radical and neutralize it before it can damage these phospholipids, right? So you can see how the free radical is damaging these phospholipids here. So if there isn't vitamin E around, it's not going to be able to neutralize those free radicals. So another sort of specific example, I do expect you guys to understand this in terms of the specific function of vitamin E, okay? It's particularly going to work at the plasma membrane and particularly going to protect those phospholipids by neutralizing free radicals that will damage them, okay? Okay, let's talk a little bit now about environmental factors that will cause cell injury, okay? And so there are various environmental factors that can cause cell injury. We're not going to talk about every single one, but we'll talk about two main ones here in sort of in detail, okay? So the main adverse effect these environmental factors will have is respiratory impairment, right? Mostly by inhaling some of these things, right? They'll irritate cells in the airways, they'll cause inflammation, oxidative stress, bronchoconstriction, also increased mucus secretion, and then obviously that will all affect respiratory function, okay? The most at-risk individuals, right, in terms of these environmental pollutants essentially affecting, respiratory system are those individuals that already have an underlying respiratory disorder or a cardiac disorder, okay? Some of the contaminants that exist in the air are carbon monoxide, lead, nitrogen dioxide, sulfur dioxide, and ozone, okay? And a lot of these are produced by the burning of fossil fuels, okay? We will particularly talk about carbon monoxide and lead in a little more detail, okay? So let's do that. So carbon monoxide poisoning, we've probably all heard of this before, right? One of the reasons why it's so dangerous in terms of how it actually compromises the function of the cells, and we'll talk about that. But the other thing is that it's tasteless, it's odorless, it's colorless. So unless you have a carbon monoxide detector around, you may not even know that you're being exposed, okay? And it can cause acute poisoning, right? It causes more than half of all deaths from poisoning worldwide. Okay, so that's a little scary, right? So a lot of if we're calling a death, due to poisoning, more than half of those will be carbon monoxide poisoning. Okay. In terms of treatment, you obviously need to remove that individual immediately from that source of carbon monoxide, and then oxygen will be administered as soon as possible. Okay. How does carbon monoxide work, right? What happens if you're exposed to carbon monoxide? Carbon monoxide will actually bind to hemoglobin, okay? So it binds to hemoglobin itself and it forms something called carboxyhemoglobin. Once that carbon monoxide binds to the hemoglobin, it prevents oxygen from binding, okay? And so if oxygen is not able to bind to hemoglobin, that means your blood's not carrying any oxygen around, right? And so obviously that's going to deprive your body's cells of oxygen, okay? Carbon monoxide's affinity for hemoglobin is actually 200 times more than oxygen's affinity for hemoglobin. So that means even if there was oxygen around, the carbon monoxide is going to bind to the hemoglobin because it has a much higher affinity for the hemoglobin than oxygen does. Okay, and when carbon monoxide is bound to hemoglobin, even if there are some other binding sites that are available for oxygen to bind, those are often sites that are stronger. And so once that oxygen binds to those other sites, it's actually really hard to unload any of that oxygen. So basically, if carbon monoxide is around, right, in your bloodstream, it will bind to hemoglobin and it will keep oxygen from binding, okay? It will also bind to myoglobin, okay? So myoglobin is similar to hemoglobin in skeletal and cardiac muscle cells. And again, it will compromise the oxygen transport in muscle, okay? So obviously it's gonna affect cardiac function. If you look at sort of clinical manifestations that are associated with some of these different levels of carboxyhemoglobin, remember when the carbon monoxide binds to the hemoglobin, we call that carboxyhemoglobin. That's what's formed. So if you have 1 to 2%, right, no symptoms. But once you start getting increased percentage of carboxyhemoglobin levels, you can see how the clinical manifestations get more and more serious. Okay, I don't think I need to read them to you, but you guys can see here. So, I mean, again, once you're getting up to these levels here, you better immediately be removed from that carbon monoxide and be treated with oxygen to try to reverse it. Lead poisoning, okay, is another thing that I said we will talk more specifically about. This normally gets absorbed into the blood from the GI tract. Or from the respiratory tract, right? Because we said it was kind of something that's in the air, okay? Can also be absorbed from the skin. It is very toxic. We've talked about this before too. I mean, children are particularly more vulnerable to the effects of lead, but at high enough levels, it's going to affect anyone. So it will affect many organ systems. So it can actually cross the blood-brain barrier. So therefore, it will affect the brain. And when it gets into the brain, into the central nervous system, it causes hypoxia and oxidative stress. Some of the clinical manifestations, you'll see seizures, movement disorders, vomiting, cognitive issues, okay, because of the effects on the brain. In terms of treatment, again, you have to remove that individual from the source. Obviously, try to prevent future exposures. If there's acute ingestion that occurred, there are chelating medications that can be given, right? Like if you think about like charcoal and stuff like that, that will bind up that lead and then basically get it out of the body. Radiation is also something that will affect cells, right? 2 different types. Again, I think we mentioned this in our environmental factor chapter, right? Non-ionizing radiation doesn't eject electrons, okay? This is UV radiation, right? Electromagnetic fields. It can, though, be carcinogenic, right? We know exposure to UV light can induce DNA damage, can lead to skin cancers, right? Ionizing radiation, on the other hand, is something that's, a lot stronger. It ejects electrons. This is, X-rays, right? Atomic bombs, nuclear testing, radon, radium. These are all types of ionizing radiation. Now, some damage that's imposed by ionizing radiation can be reversed. But again, if it's going to cause mutations in the genome, that's not going to be reversible. Okay, so it kind of, again, depends on how much exposure has occurred. It says acute exposure to high levels. We'll look at this table that's on the next slide in a second, okay? Can lead to nausea, fatigue, diarrhea, hemorrhage, CNS damage, and death, okay? Again, if it's high enough levels, Okay, so if you look at this this table here, did I say, oh, table 10.3, let me just see something. I think I meant figure 10 point, let me just make sure I didn't mean table 10.3. Oh, I did. Yeah, so there is a table 10.3. That's what I was referring to there. So you guys can take a look at that. And maybe once I finish the chapter, I'll just pull it up just so that you guys can see it here if you don't have access to the textbook. And basically what it's showing you is basically the exposure levels, right? And what the health effects are. Again, I don't expect you guys to memorize any of this, but I think it gives you kind of, perspective in terms of, what types of levels and what types of health effects you'll see, okay? This next figure here, this 10.13, is basically kind of showing you a couple things, right? Different types of ionizing versus non-ionizing radiation, right? In terms of effects, okay? So here, again, when you're talking about ionizing, this is where you're going to see, DNA damage, okay? And again, if you're talking about, obviously atomic bomb and things like that, exposure, right? Really high levels, it's going to lead to very serious effects, okay? And that is shown in that table 10.3 that I was referring to. Okay, so I'm going to stop there. Part 2 will pick up with this learning outcome and talk about endothelial and epithelial cells particularly, okay? Let me just pull up that, that table just so that you guys can see that real quick. I'll pull up that table just so that it's at the end of this lecture so you know what I was referring to, okay? But part 2, again, we'll pick up at learning outcome 10.5. Okay, so just to show you that table that I was referring to, one, I just want to show you this as well. So remember we said, I talked about lead, and I also mentioned or reminded you that, children are particularly vulnerable, right, to the effects of lead exposure. And so if you want to take a look so these are different blood levels of lead in children, and you can see what effects these levels have as you get to increasing levels right here, okay? So starting at 10 micrograms per deciliter all the way up to 150 micrograms per deciliter, okay? Let me also show you that figure, that table 10.3, which was referring to really the effects of acute exposure to high levels of ionizing radiation, okay? So these are exposures that are not normal, right? That we're going to basically be exposed to if you get up here during regular life, essentially. So if you talk about REMS, so that's one of the ways that you basically measure the amount of radiation, okay? And so you can see as you increase here, you can see some of the health effects that will occur, okay? And so just to kind of, again, give you an idea of the sort of varying degrees of intensity in terms of those health effects. All right, that's it, guys. Like I said, part 2 will pick up with 10.5, which is effects on endothelial and epithelial cells. All right, thanks.